Another solution is to fit a model other than the Poisson regression model when we know the Poisson distribution is not correct. The negative binomial distribution is often used when your count response has over-dispersion. If F has a gamma distribution, then Y sub I follows a negative binomial distribution. Assume we are interested in the intercept-only model and that mu sub I is distributed gamma. Then the expected value of mu sub I equals mu, but the variance of mu sub I is equal to alpha times mu squared. So there are two parameters in our negative binomial regression model. The negative binomial distribution is modeling the number of successes before R failures when the success probability is P. For example, when flipping a coin, how many heads will you get before getting 3 tails? The expected value of mu is still mu, but the variance is something greater than mu. So we call alpha the over-dispersion parameter. When alpha equals 0, the negative binomial distribution degenerates into the Poisson distribution, so the negative binomial is a generalization of Poisson. We will fit a negative binomial model where the regression formula looks essentially the same as the Poisson model. The interpretation of the model parameters are the same as in Poisson regression. The difference is, instead of assuming Poisson, here we assume that Y follows a negative binomial distribution. So we have this extra over-dispersion parameter alpha. The only assumption that changes is that Y follows a negative binomial distribution with over-dispersion parameter alpha. Otherwise, we still assume that the log of the expected value of Y given X is a linear function of our predictors and that the values of Y are independent. In order to test for over-dispersion, we can use the boundary likelihood ratio test to assess whether there is truly over-dispersion in our count data. If there is no over-dispersion, it is more efficient to fit a Poisson regression model because it has one less parameter. So we want to test the null hypothesis that alpha equals 0 against the alternative that alpha is greater than 0. We compare the log likelihood of a model with alpha and another model without alpha. The test statistic is given by this equation. The p-value is 1/2 of the probability that a chi-square value with 1 degree of freedom is greater than the observed test statistic. So instead of comparing to a chi-square distribution with 1 degree of freedom at the 0.95 level, we use chi-square at the 0.90 level because alpha can only be greater than or equal to 0. So our alternative is alpha is greater than 0. The function for fitting a negative binomial model in R is in the mass package, which should be included in your base installation of R. After fitting the negative binomial model, we perform the boundary likelihood ratio test, which results in a very small p-value. So we conclude that alpha is non-zero, and thus a negative binomial model is preferred to the Poisson model. When testing whether age is significantly associated with the reads for this gene, we fail to reject the null hypothesis. This conclusion is concordant with the conclusion reached when using the robust standard error estimate. DESeq and EDGE-R are bioconductor packages designed for analyzing RNA sequencing data. Both are based on the negative binomial distribution and were initially designed to compare a small number of samples. This was due to the high cost of sequencing, and therefore both methods use some approach for sharing information across genes, which is similar to lemma. EDGE-R uses an empirical-based procedure to shrink the gene-specific dispersions toward a common dispersion, which are then used in fitting the negative binomial model to each gene. Differential expression is then tested using standard likelihood ratio tests. After model fitting, the resulting p-values are used to typically estimate the false discovery rate. The DESeq package was subsequently upgraded to DESeq2, which is also based on the negative binomial distribution and also expects the raw counts of sequencing reads are provided. Each sample should be an independent sample and not technical replicates. If your experiment includes technical replicates, which is when the same library preparation was distributed over multiple lanes of the sequencer, that is, the sample is divided and hybridized more than once, the resulting reads from technical replicates should be summed, forming one unique sample. Y sub ij is the read count for gene J in sample I, which is assumed to follow a negative binomial distribution with mean parameter mu sub ij, which is the mean count for gene J in the sample I, taken to be the quantity q sub ij, which is proportional to the true concentration of fragments from gene J in sample I, scaled by a normalization factor S sub ij. In most cases, S sub ij is equal to S sub I. That is, the same constant, S sub I, can be used for all genes in the sample I. The negative binomial model also includes a dispersion parameter for each gene, alpha sub j. The linear part of the model assumes that the log of q sub ij is a linear function of the predictors. In the DESeq2 package, the log base 2 link is used rather than the natural log to facilitate interpretation as a fold change. In fact, DESeq2 also uses empirical Bayes to shrink the log fold change estimates. The Wald test is based on the shrunken log fold changes and their standard errors for testing differential expression as the default inference method for extracting p-values. Thereafter, genes are typically identified using a suitable false discovery rate threshold. As a first processing step, the effective library size is estimated. The normalization constant, S sub ij, is similar to the library size and controls the amount of biological aliquot loaded for each sample. By default, S sub ij is equal to S sub I, which means that all genes in the sample are normalized to the same constant. This normalization constant is formed by taking, for gene J, the geometric means of counts for that gene across all samples to serve as the reference. Then for sample I, we take the ratio of the jth gene count to its reference. Once these ratios have been formed for all genes in the sample, then we take the median for all genes having a non-zero estimate for the reference geometric mean. The median is robust, so it is not affected by outliers, but is somewhat comparable to the library size. Alternatively, gene-specific normalization factors can be used to account for technical biases, such as GC content or gene length. We may optionally remove genes that have very low or no counts across all samples. As a reminder, for Lima, we fit a model and often examine contrasts of interest. When using DESeq2, we can also examine contrasts of interest as our hypothesis tests. In Lima, we moderate the variance of sigma squared, but in our negative binomial model, alpha is our dispersion parameter, which relates to the variance estimate. Therefore, in DESeq2, we use empirical Bayes to estimate the dispersion parameter to avoid underestimating alpha sub j. We first load the DESeq2 library. We want to use DESeq2 to compare these three groups represented in call data sample type. But DESeq2 cannot use character string data, so we first create a variable group to be a factor representing primary, recurrent, and normal samples. Then we set up our DESeq dataset using the DESeq dataset from matrix function. DESeq2 is an object-oriented package that expects the count data to be in a count matrix, and call data is the phenotypic data object as in the summarized experiment package, and as such expects the number of rows to equal the number of columns in the count matrix. Here, it is best to use call data associated with your count data matrix to ensure the gene and phenotypic data are aligned. Then the design matrix is specified. As usual, tilde represents a regression model where the predictor variable is on the right-hand side of tilde. Type the name of the object to see the object to verify the dimension, column names, and the information on the samples. Then after constructing the DESeq dataset, we issue one command, DESeq, which adds more information to the dataset. It first estimates size factors, then dispersions, the gene-wise dispersions, the alpha trend function, which is the mean dispersion relationship, then estimates map for all alpha parameters. These are used in model fitting and testing. As I mentioned, for outlier genes which have large dispersions, DESeq2 uses the gene-wise estimates and re-estimates those models. That is, it replaces the shrunken dispersions with gene-wise dispersions when it is considered an outlier and refits the model again. You have the option of decomposing DESeq into these three steps: estimate size factors, estimate dispersions, then fit negative binomial models. But unless you want to add your own functions in between these steps, you may as well use the single DESeq function. To get the results, suppose you want to examine the contrast of primary versus recurrent tumor. We can apply the results function and pass our DESeq data set and specify the contrast we want. Because the design matrix may include more than one covariate, on the right-hand side of the model formula, as a predictor, we do specify the name of our predictor variable. When we control the false discovery rate at 5%, we get 397 up and 793 down-regulated genes. We can sort the resulting object by FDR, which is in the ADJP for adjusted P column, and examine the top few genes. We can also construct a volcano plot. There is no function in DESeq2, so the code is provided here. We are simply plotting minus log base 10 of the false discovery rate against the log base 2 fold change and requesting that genes having an FDR less than 0.05 and a log 2 fold change greater than 2 be represented in blue. Otherwise, they are represented in gray. The horizontal line corresponds to an FDR of 0.05, such that points above this horizontal line represent genes significant at the FDR less than or equal to 0.05 level. The vertical lines represent -2 and +2 log 2 fold change. Beta sub JR represents the log base 2 fold change for gene J and a 1 unit difference in covariate R. But this log 2 fold change is not useful for low reads. For example, if you have a sample with 2 reads versus 1 read, the log 2 fold change is 1. But this is likely due to randomness. If you have a sample with 200 reads and a sample with 100 reads, the log 2 fold change is also 1, even though the stability of the estimate for the first example is worse than the second example. We therefore want to pull the estimates towards 0 for low count genes, and we can do that using empirical Bayes, in which case a prior is put on the coefficients, except for the intercept. This could be an adaptive student's t, normal mixture, or normal prior. The latter is no longer recommended. Map estimates of the coefficients are shrunken towards 0, especially for low count genes. This shrinkage can also be applied to the contrasts. Here is the MA plot for the primary versus recurrent comparison, where the unshrunken results are plotted. The y-axis is the log base 2 fold change, and the x-axis is the normalized counts. If the false discovery rate is less than 0.05, the gene is colored in blue. To the left are low count reads. You can see strong log base 2 fold changes in that region, which we do not want. The DESeq2 package includes the LFC shrink function, which adds shrunken log 2 fold changes and their standard errors to a results table from DESeq run without LFC shrinkage. ASHR is an adaptive shrinking estimator that assumes a mixture of normals prior and can be applied to either coefficients or contrasts. When shrinking the log fold change, we pass the DESeq data set and tell it which contrast we want to shrink and what type of shrinkage we want to do. In this case, ASHR, again, for adaptive shrinking estimator. Notice in the resulting MA plot that there are no longer high fold changes for low count genes. Now the significant genes have higher normalized counts. When examining the primary versus recurrent results when the log fold changes are unshrunken, to the same comparison with when the log fold changes have been shrunken, we see the table reports the same base mean, p-value, and FDR. Only the log fold changes have been shrunk towards 0, and correspondingly, the standard errors increase. The test statistic is the log fold change divided by its standard error and is removed from the shrunken results because the test statistic is from a different model. Another shrinkage method is APE-GLM, which assumes an adaptive student's t prior. Here you can only use the coefficients and not specify the contrasts, and then specify the type of shrinkage, in this case APE-GLM. As seen in this MA plot, This method seems to shrink better. So the limitation here is that APE-GLM only applies to the coefficients and not to the contrast. So it is important to denote which reference group you're using when you have multiple groups in your linear model. A few comments on filtering. Some genes have many zero counts in many samples, or low expression across all samples, or they may have low variance. Some researchers prefer to remove these genes or even genes having low variance across all samples. This reduces the number of multiple comparisons. However, you may miss some truly differentially expressed genes that have low expression levels. Likely, filtering out low count genes is the best approach. That is, it is better than simply applying a low variance filter. Just be sure that your filtering criteria do not depend on the phenotypic variable of interest. Here are some useful references for further reading. Additionally, the Bioconductor package vignettes for Lima, DESeq2, and EdgeR can be explored.